Have Allison back in the MVZ. So Allison graduated from Cal in 2007, and she was involved in the MVZ in the early days of our undergraduate program. Allison participated in several Grinnell Resurvey Project trips up in the Lassen area and Southern Sierras. She learned to skin birds in the field there. Also did some shooting with us, remember that. And Roy mentioned her honors, so she was an honor student, so her honors thesis was looking at a century of avifaunal change in birds of the Berkeley campus, and she published that in the Condor with Rory and Morgan Tingley, also a former PhD student, in 2008. And she also updated this publication that was first published in 1922, list of the birds of the Berkeley Hills, including the Berkeley campus. I have a number of copies. I found them in my office as I'm cleaning things out. So if anybody wants one, I have them here. So it's kind of a classic. And another fun fact, Allison was senior manager of the Cal Band when she was here. So she left Berkeley, and and one more other thing that she did when she was here was she was the coordinator for our centennial event, which was a huge event in 2008. So Allison was really involved in the MVZ. She left in, what year did you leave? 2008, okay. To go to San Diego State University to do a master's with Kevin Burns, also former MVZ alum. And then from there she went to Harvard to do her PhD with Scott Edwards, also former MVZ alum. And Kevin got his master's with Bob Zink, another MVZ alum. So lots of MVZ connections here. And about 6 months ago started her dream job as assistant curator at the LA County Museum of Natural History, where she is today. Her three research areas, if you look at her website, are host-pathogen coevolution, the evolution of plumage coloration, and the genomic impacts of introduced species. And so it's great to have Allison back, and today she's going to talk about her research. What she's done since.
It's really fantastic to be back here to see some familiar faces, some friendly faces, and a lot of new faces as well. You know, the MVZ really was kind of what shaped me. I didn't really know about birds or field research or anything like that till I got involved here and took IV 104. So, and you can see it's stuck, right? So that's great. So, you know, as many scientists here in the room are true, so one of my goals as an evolutionary biologist is to understand what the processes that generate the amazing patterns of biodiversity we observe on Earth. And so there are several different approaches to doing this. One approach is to study a number of different species and to look, you know, take a trait, look at how species with this trait with different evolutionary relationships are correlated in terms of abiotic or biotic factors and make predictions as to, you know, what might be driving the evolution of this trait. And on the flip side of that, there's another approach that people take to really get at the processes, which is difficult to do when only you have this kind of long-scale data, which is actually to look over time at how traits change. So we can actually observe kind of in real time how the process of evolution is happening. But it's difficult to do this in kind of non-experimental context, except in a place like a natural history museum, where we have kind of series through time. So in my own research, I try and take advantage of both of these different types of timescales and kind of integrate to study the patterns as well as observe the processes that produce them. And so to try and really connect how are these patterns of biodiversity formed. I also study another axis, which is kind of the phenotype-genotype axis, like I know many people here are interested in, but what's the genomic basis for producing these traits that we observe? And so my research kind of falls in different places. I'm going to talk about today, just briefly introduce one of my main research topics, which is plumage coloration, which kind of is spanning more of kind of this phenotype-genotype axis. So far, I have studied the tanagers and looked at across this big tree how is plumage coloration changed, kind of from a visual perspective. And I'm now kind of going deeper into that axis towards genotype, but stopping at the mechanisms, so digging into what are the actual coloration mechanisms producing these changes, and eventually go into kind of what are the genotypes. But today I'm going to focus more on a different aspect of my research program, which kind of takes more of a genotypic approach, but is more of the time series spanning. And I'm going to connect kind of this interspecific view with these time series with population genetics at different scales. And so the system I use for this is host-pathogen coevolution. Because, so here's my little virus here, and here's my bird. So viruses are constantly, or pathogens generally, are constantly evolving at being better at infecting their hosts, and hosts are being better at evolving resistance to these pathogens. And so, over a long-term time scale, this can produce patterns of coevolution, often termed the Red Queen hypotheses. And getting at what these actual processes are is tricky. Many people use experimental approaches to understand, how are these animals being resistant? How is that happening? However, in natural systems, sometimes it can be difficult to do experimental approaches. So I'm taking more of a genotypic approach to understand, to actually look at what are the patterns of these processes that we observe in the genome. So over long time scales, I'm using comparative genomics, where we compare the genomes of many different species. And we can do this both to infer the relationships of these organisms, but also to look at where positive selection has occurred. I also am using a population genomic approach. You'll see a couple different projects there to say kind of in the population level, do we observe different signatures of selection? We know from studies of humans that pathogens are probably one of the most important drivers of kind of genetic variability in human populations and one of the strongest selection factors. However, many non-model organisms have really restricted their studies to kind of some of the classic immune loci like MHC, which of course is very important, but there are many other aspects, there are many other types of immune loci that we can study. And so what I'm doing here is I'm going to use a whole genome approach at these different time scales to really look at, beyond just MHC, how are pathogens shaping bird genomes? So the first question I'm going to ask is, what are the genomic consequences of pathogens over long time scales? I'm going to kind of start at the longer end of the spectrum. And as I mentioned, so my model system are birds, and birds are excellent models for studies of disease, ecology, and immune system evolution. Here I've got just a kind of rough cartoon of migratory pathways. So we know that birds migrate to very different parts of the earth, which means that they're constantly mixing different pathogens, which means that they're experiencing different pathogen communities. Birds are also important in the poultry industry and have the potential for some of their pathogens to spread to humans. So we know quite a lot about the avian immune system compared to some other types of non-model organisms. And so that means that we actually know what some of these genes are and how they're functioning in birds. Birds are actually also excellent models for studies of genome evolution. As of yesterday, when I checked, there were 141 bird genomes available on NCBI and more added all the time. They also have a very small genome compared to mammals. It's about the third the size of mammals, about 1.2 gigabases or so. About 38 pairs of linkage groups, including these macro chromosomes, these longer chromosomes, and these very short microchromosomes, and the very high levels of recombination, which means that in organisms like humans, oftentimes when you're trying to identify what part of the genome is under selection, it'll be a region that involves, say, hundreds of genes. It'll be this big chunk. But in birds, because recombination is so high, we actually can pinpoint pretty specifically, what regions under selection and what single gene or what set of genes might be associated with that. Okay, so birds are great for many things, many other reasons as well, but we don't have to go into that now. But let's ask the question, do pathogens target the same genes? So here I've got my immune system pathway with our recognition genes, some signaling genes, and some effector genes that are going to affect it. And so, what's one possibility is that host pathogen hotspots are shared, which means that you might have, across different species and different pathogens, they might be targeting the same genes over and over again. So there might be kind of these hotspots of interactions where we see this kind of coevolutionary arms race taking place again and again. Another possibility is that host pathogen hotspots are species specific, which might mean that for each different species and maybe for each different pathogen, there's kind of a single gene or a single protein that they're targeting in order to subvert the immune system. So it might be very species specific in terms of which gene is under selection and ongoing these kind of host pathogen evolutionary arms races. So what I wanted to do essentially was to test for these two different possibilities. And to do that, I did a scan for positive selection. So I wanted to test, okay, let's sequence all of these different genes. Which genes are under positive selection across a large number of species? To do that, I estimated a ratio called omega, which is the ratio of non-synonymous substitutions, or substitutions that actually change the amino acid and change the protein, to the number of synonymous substitutions, which mean that these are substitutions that actually have no sort of change in the amino acid or the protein. Under neutral evolution, we expect this value to be 1, so we don't expect there to be any difference in the number of non-synonymous or their synonymous substitutions in the ratio of those. Under purifying selection, we expect there to be fewer non-synonymous substitutions, and most genes are under purifying selection because generally when you break a protein, it's going to be bad, it's not going to be good. However, some genes, or some portions of some genes, may be under positive selection, which means that they may have more non-synonymous substitutions compared to synonymous. And so essentially what we were doing was testing for, not necessarily across the whole gene, but across parts of the genes, do we see evidence for some set of sites that might be under positive selection. And we did this with a rigorous model testing approach. For those of you in the know, using a bunch of camel models and also the HiFi package with Busted and BSREL. So I analyzed about 11,000 protein coding genes from 39 species, look for signatures of natural selection in birds. For those of you who are familiar with birds, these kind of generally span the bird phylogeny pretty well, and found that about 10% of genes actually show evidence of strong positive selection across birds, which is pretty high. So we were kind of surprised by that. But now we said, okay, well, are these genes kind of clustered in with any sort of functional annotations? So we looked at KEGG system pathways, and basically, so if here are 4 kind of cartoon pathways, let's say these pink genes are the ones under positive selection. Did any pathways have more genes under selection than expected? And so, for example, in this case, we would say this immune system pathway was enriched for positively selected genes. So we did this with all of the functional pathways that we know for birds, using the KEGG database. And we actually did find a number of pathways that were enriched for positively selected genes. Now in this graph, what I'm showing here is each of these darker colored circles represents a pathway. Each lighter colored circle represents a gene that's connected to that pathway. So you can see some genes like this one are shared by multiple pathways, but a number of them are only part of a single pathway as well. So it wasn't necessarily, the signal is not driven by a few genes that are in a bunch of pathways, but it's kind of a mix of shared and individual genes. And we noticed that there were essentially kind of 4 main functional categories that had enriched pathways. The blue, the biggest one here, are pathways that are involved in the immune system in some way, either through infectious disease and signaling molecules and interactions. So this was exciting because it suggested that what we had hypothesized, that these pathogens are kind of targeting the same genes across different species, because remember, these are genes that are under selection generally across all birds, is true. But interestingly, we found a few other types of pathways as well. This kind of second biggest cluster was replication and repair pathways. And this, we were a little bit confused at first, but then it actually made sense when we thought more about it, because one thing that makes bird genomes so small is they don't have a lot of repetitive elements and transposable elements. And it's actually been hypothesized that homologous recombination is essentially the immune system for transposable elements. And so the fact that we're seeing that same kind of signature of positive selection might suggest that there's actually some sort of arms race going on between transposable elements and recombination machinery in birds as well. And so we observed this. Also, it's kind of hypothesized that flight and the very strong metabolism that's associated with being able to fly is also a strong selective pressure. And so some of these like base excision repair and mismatch repair is hypothesized to be driven by this kind of strong metabolic need as well. So that was kind of cool to observe. We also, a couple of pathways in lipid metabolism, which is important for flight as well. And finally, phototransduction, which is involved in the visual system, which is of course very important for signaling in birds. So this was all very cool, and suggested that, you know, overall the majority of the signal we saw was probably due to host-pathogen kind of co-evolutionary arms races, either your traditional pathogens or more kind of genomic pathogens. However, we wanted to test this in another way, and not just rely on these kind of KEGG category classifications. So we also pulled down all transcriptome data that we could find in publicly available databases for birds that had been infected by some sort of pathogen and had transcriptome data available with infected and uninfected birds. We found in the end 12 different studies that had been infected by a number of different viruses on the top, bacteria or a protist on the bottom. And what I'm going to show you here are the proportion of genes. We classify genes as being upregulated in response to pathogens, downregulated in response to pathogens, or not differentially expressed in response to pathogens. And then we estimated the proportion of genes in each of these categories that were under selection in birds. And so what do we see? Well, we see that basically for all different pathogens, there were more genes in the being upregulated and under selection in birds compared to genes that were not were downregulated or not differentially expressed, except for kind of Plasmodium here, which it was not super strong signal, but also had downregulated genes generally under selection. So that was West Nile virus, but that's only 4 genes, so it's, you know, hard to say how true that is or not. But overall, this corroborated our kind of hypothesis that we had built and what we had observed with the KEGG pathways, that pathogens are really important drivers of selection across all of the birds. Now, so far I've only been looking at signatures of selection that were true across birds, but also it's possible that there were other genes that might be under selection only kind of in different specific lineages of birds, perhaps different lineages that shared certain life histories, for example. For example, maybe migratory birds or birds with different social structures. So we also used a program called BSREL to actually estimate genes that were under selection in each different bird lineage. And I conducted a principal components analysis of the omega values, the strength of selection, and the proportion of genes under selection across these species to see if there were certain evidence of kind of suites of genes under selection in different lineages of species. So this is that principal component 1 actually had kind of different sets of species that were not each other's closest relatives under selection together. And so if you kind of look, you've got some ratites up here, under also penguins and eagles were all kind of shared genes under selection. Passerines down here, as well as hummingbirds and swifts and doves. And so we were kind of thinking, what are life history characteristics that might join each of those different clades? And so what it turned out is that one of the kind of basic proxy for some different types of life history is body mass. And so when I actually regressed this PC1 against body mass, they were highly correlated. So essentially we're seeing species with similar body masses having these sets of genes under selection in those different lineages. So you might wonder, what's actually driving that? Well, I did then did a gene set enrichment to understand what those genes were with this correlation between body mass and PC1. I found two pathways that jumped out, which were FOXO signaling and cellular senescence, which are actually both related to lifespan. And so body mass is a proxy for lifespan, so actually what we're observing is probably not pathogen related, but it's actually probably lifespan is the thing that's generally bringing together signatures of selection in these different species, at least the strongest kind of signal that we said. So that was pretty exciting. So kind of coming back to our question, we found that immune genes are enriched for signatures of positive selection. And we found that genes upregulated by pathogens are more likely to be under positive selection. And finally, genes under selection in only specific bird lineages may be related to life history, which is body mass or longevity. And so not necessarily certain kind of pathogens or life history strategies, but actually longevity may be that driver. And so this was all great. And so, you know, kind of this was great about birds, but another study had been published in the meantime looking at genes under selection in 24 different species of mammal, and found that virus interacting proteins that interacted with viruses in some way, were 30% had signatures of adaptation 30% more strong than other genes, and also found that these parasite interacting proteins, parasites being things like malaria, had 3 times more adaptive substitutions. So we thought, I wonder if some of these same genes are under selection in birds and mammals. And so luckily about 5,000 genes in our data set and this data set overlapped, and they'd used the same methodology, so we were able to kind of directly compare them with our own data. The first thing we wanted to know is there an overlap in genes under selection in birds and mammals? So we just could have, at different FDR cutoffs, we did this odd ratio test where kind of this red line is there's overlap is as expected. Above the red line means there's a greater overlap in both clades. Under the red line is less overlap. And we found that there is more overlap than expected at the highest FDR cutoff, and kind of continues no matter what FDR cutoff we use for considering a gene to be under selection or not. So this is exciting. This means that genes under selection in birds are more likely to be under selection in mammals. But what about the functions of these genes? So again, we did a pathway enrichment, this time using as the background, instead of all genes as the background, the set of genes under selection in birds, and kind of as our test case, genes under selection in birds and mammals. So we wanted to ask, you know, compared to genes under selection in birds, were perhaps the pathogen interacting genes more likely to be under selection in birds and mammals? And what did we find? Well, these are essentially kind of null distributions with our empirical estimates of the enrichment percentage there. And we find that in 2 cases, that pathways were enriched for genes under positive selection in birds and mammals, and those were influenza A and herpes simplex infection, so both virus pathways. And so this kind of suggests that it really does seem like viruses might be targeting a lot of the same genes kind of over and over again in birds and mammals. And finally, to kind of just, you know, top this off, we pulled 14 studies that had transcriptome data available for mammals that had been infected with one of the same pathogens as used in one of those bird studies. And so we had data for E. coli, influenza A, mycoplasma, plasmodium, and also West Nile virus. And calculated the odds ratio, so do we see the same kind of expression patterns due to these diseases in birds and mammals? So for example, here we see that upregulated genes are more likely to be upregulated in both clades than expected, but downregulated genes are not for E. coli. And this is true for influenza, for mycoplasma, but interestingly not for plasmodium. So there we see more downregulated genes similar in both clades. There might be something interesting going on there. But finally, okay, so we know that genes are likely to be under selection in both clades. Genes are likely to be kind of up or down regulated the same way, upregulated the same way, you could say, in both clades. Are there genes that are upregulated and under selection in both clades? Well, there wasn't enough statistical power to detect that with a model, but we did find 5 different genes that did fall in that category. And kind of interestingly, PKR is kind of the example gene for host-pathogen coevolution, studied in a lot of the medical literature, for example. MX1 is known to interface directly with influenza, so it's kind of also evidence for direct influenza interactions. But these 3 haven't really been studied extensively kind of in either the medical community or the biological community. So I think these could be kind of good possibilities for some future work in studying, what specific genes are driving a lot of these kind of host pathogen coevolution, these arms races. So coming back to our conclusions from our kind of long term, we've gone even longer term or deeper in the evolutionary relationships and found significant overlap of pathogen-selected genes in both birds and mammals. All right, so we've kind of hung out on this interspecific side of the coin here. I'm going to come and look at some population genetics. And so this is some pretty preliminary work that I started last year when I was my very brief time as a bioinformatician at Harvard. But basically, I wanted to look across bird populations. Do we see the same genes under selection at the population level as we see over these long-term timescales? Which is what I just said right there. And so there's a huge volume of population genomic sequence data publicly available on NCBI that we could make use of. But very little of it is available in processed and kind of analysis-ready form. Here's just, so birds are up here. There are 32 different species with data sets available across the tree, many others as well. And so the first thing I did was to build a reproducible and optimized pipeline for non-human population genomics, which involved kind of automated the process of pulling down reads from NCBI, mapping, doing variant calling, and filtering. So I'm hoping to get this out kind of next year for any of you who are interested in using NCBI. We're going to make all of the VCFs available too in a database for downloading, which is very exciting. But from a biological perspective, how did I estimate selection? Well, it's kind of with the same idea as that omega value, but with the McDonald-Kreitman test, where we're looking not just at the number of non-synonymous or synonymous substitutions, but of the proportion of fixed non-synonymous and fixed non-synonymous substitutions to polymorphic non-synonymous and polymorphic synonymous substitutions. So under neutral evolution, we expect this ratio to be the same. So this is all in a population compared to some outgroup. Of course, under negative selection, we expect there to be fewer non-synonymous to synonymous fixed polymorphisms compared to polymorphic, and positive selection is the same. So again, for coding genes, we're looking at any that we can find that are under positive selection. So I have about 30 species run through my pipeline, but haven't finished yet all of the MK tests and kind of analyzing that data. So I'm only going to show you some preliminary results for 3 different species. Gallus varius, the green jungle fowl, a relative of our friend the red jungle fowl, or the chicken. European flycatcher, and then zebra finch. And so, interestingly, positive selection was only found about 1% of genes, so it's interesting to think about kind of power for these types of MK tests versus something like kind of model fitting with PAML. But the first thing I want to know, okay, were there any genes under selection in all of these species as well as my overall bird data set? And in fact, there were 3 genes that were. And interestingly, this PRAP9 gene was also one of the ones that was under selection in both birds and mammals and upregulated in pathogens to both birds and mammals. So we see that popping up again in this population data set. And these 2 are either genes that are involved in replication or the immune system. So it does seem like there might be something to this and might be some signal in terms of kind of population genetic processes producing these long-term signals that we observe. Also, I estimated what's the proportion of overlap in the genes under selection in each of these species compared to just null random expectations. And we found much, much greater overlap between 40% and 60% of genes were under selection in each of these species and in my genes under selection across birds compared to expected. So again, it does seem like it may be these population level processes kind of adding up over time that might be driving that pattern that we observe, which is very exciting. So unfortunately, this is all I can show you on this today, but kind of stay tuned. There'll be more coming in the future. So for this question of does selection at the population level contribute to signatures of positive selection at longer time scales? Well, we find that there's a significant overlap in genes under selection across birds and in several bird populations. And we find that at least some immune-related genes are under selection in all bird populations. So more on that soon. But in the meantime, I'm going to step forward to kind of the most recent time scale I work at, which is time series. And I'm going to ask the question of what are the consequences of a novel pathogen on a host genome? So can we start to see some of this process that we've been observing at longer time scales in real time? And so for that, I use the House Finch as a model system, which is really a model for studies of ecology and evolution. And from a disease perspective, it's interesting because house finches were exposed to a poultry-associated Mycoplasma gallisepticum. It's a bacteria. In 1994, there are western and eastern populations of house finches. Unfortunately, I don't have time to go into all of those interesting dynamics, but happy to talk about that later if anyone's interested. But what this mycoplasma does in house finches, which it doesn't do in chickens, is it gives them conjunctivitis. This is actually a bird that I captured in Massachusetts. And you imagine if you're a little songbird and your eyes swell shut and you can't see anything, your survival odds are probably not great. So in fact, that was true because within about 2 to 3 years, it spread throughout this entire eastern population. And in many areas, we saw about 60% declines. So the disease had a huge impact on house finch populations. It took about 10 years for it to kind of get to the western population, but it's been spreading pretty rapidly ever since. And I think there's some kind of, we don't really know what's going on in the southern part of the range, but there are kind of some current outbreaks of it happening as well. So what we do know from work that had been done in the Edwards lab previously, where they had collected some populations from Arizona before Mycoplasma got there, so these populations had never seen it before, and some from Alabama that had lived with Mycoplasma for at least 10 years. They've been kind of evolving together. Infected them and observed what did they have in terms of resistance and tolerance. And so what they found is that, in fact, Alabama populations were much more resistant to Mycoplasma, so this kind of 10 years that they've been infected, at least it seems like they've evolved resistance. And they'd also evolved tolerance, which means for a given amount of Mycoplasma, they showed less swollen eyes. So kind of those symptoms that made them under selection the most, that really was the predation, also occurred. So this suggests that there's been evolution in this very short time scale due to this Mycoplasma disease. So what I wanted to do was to make use of museum samples to collect populations from before Mycoplasma and then after Mycoplasma, so we can kind of do some time series analysis to understand how evolution has occurred. And of course, I don't need to tell anyone here that museum collections are windows into the past, and so I was able to use a series of tissues, I'll show you here, collected from throughout the house finch range by one of our collaborators in 1990 in the western population, and in 2001 in the eastern population, 2001 in the western population, which were both before Mycoplasma in each place. Other collaborators had gone out and collected blood samples here 10 years after. I went out here, which in 2014, I came to the MBZ, I skinned a bunch of birds. This was the last time I was here. It was very fun. Also to Washington. Then I also collected some populations 20 years after, so we could really see kind of what's happening through time. So I did a low coverage whole genome resequencing for 216 house finches, as well as closely related purple and Cassin's finches, and some outgroups of other types of finches. And so in the end I had, because of this kind of 10 years and 20 years after, in the east I had 3 different time periods, in the west 2 different time periods. So I'm just going to discuss 2 different questions today, which is what genome-wide effects did the epizootic is like epidemic, but of animals, have on genetic diversity? And how did house finches evolve resistance and tolerance? So here I'm showing pi, which is a measure of genetic diversity. Each kind of circle is one of my subpopulations. And here we've got western pre-mycoplasma, eastern pre-mycoplasma. Again, you notice eastern is lower. That's due to the introduction, but a whole nother story, which I don't have time to get into. But in both populations, 10 years after, we do see a minor decrease in genetic diversity of about 1 to 2%, which doesn't sound like a lot, but from a population genetic perspective, it's actually pretty surprising to see any decrease in genetic diversity. And so what this suggests, which had kind of been recorded kind of from bird surveys and things, was that there was a lot of kind of local extinctions. And so we see kind of locally many populations going extinct or extirpated, which means that we're losing those chunks of genetic diversity. But interestingly, 20 years after, some of that had kind of gone away, which I think means that after enough time there was enough kind of mixing to kind of regain that genetic diversity again. So interesting to kind of see how that progresses into the future. I think it'll be cool to see what happens. But so what we do see is we saw a minor impact on genetic diversity, a minor kind of bottleneck due to this mycoplasma epizootic. But how did house finches evolve resistance and tolerance? Well, first let's talk about how a population can evolve resistance and tolerance. One possibility is a selective sweep of some kind, hard or soft, which is a change in allele frequency at a single locus. So for example, let's say this orange bird has a C here instead of G and is resistant. Well, kind of over time, we would expect that C to be percolated throughout the population, but also the different SNPs that were linked to it. So this produces a pattern that we can make use of later. But just kind of from a basic perspective, if I was just to look at the difference in allele frequencies, the difference in the proportions of, say, C's and G's, before versus after the selection event across the genome, and if it was just one locus, I might expect to see some sort of spike in differences, but kind of a low level of just general, very few differences. Another possibility is polygenic selection, which means there's a change in frequency not at a single locus, but at multiple alleles that would already be present in the population. And so in that case, we see, kind of a bunch of small peaks, but much reduced because that signal is going to be much weaker. So what did we see in house finches? Well, this is my kind of similar type of graph. We've got genome position on the x-axis, minus log p-values. This is just a measurement of how different they are. We call this line the line of statistical significance after multiple test correction. And we see generally not a lot of, unfortunately, we don't see a big spike. Of course, as a grad student, you see this and you're like, darn, oh well. Anyway, it's okay. Interestingly, we do see the same thing for Western or Eastern and in these different time series that I tested. If we look at FST, which ranges, of course, from 0 to 1 through time, we do see that FST is very low, but there is a little bit of a tail in each of these populations that goes out. So we do see some loci that do seem to be differentiated through time. And so, one thing I then did was to look at, okay, if we look at that top 0.1% of FST, this is just in the East 10 generations after, 20 generations after, one scaffold that kind of had the most distinct, the biggest cluster of these top 1% of FST bubbles. Well, we can actually bring in other types of population genetic parameters, not just differentiation, to make use of finding these signals. So this is a measurement called R-squared, which is a measurement of linkage disequilibrium. And so remember when that selective sweep occurred, it kind of pulled along the SNPs that were next to it. Well, when it pulls along, that's going to increase linkage disequilibrium. So if I measure that for my 3 different time periods, kind of one cool thing I found was in this peak here, we see increased linkage to equilibrium in 2001 and 2014, my 2 post-epizootic compared to my pre-epizootic kind of lower level. And so, you know, these types of regions occur in about 8 different scaffolds. And now what I'm doing is, I unfortunately don't have the results to show you yet, but actually using machine learning to make use of not just 1 or 2 summary statistics, but using a set of 10 or 12, and actually using simulations to estimate what, given the demographic scenario that I have, we estimate regions that are neutral with a soft sweep, with a hard sweep, or adjacent to these sweeps, and use these simulations to create a training set that we can then use to actually kind of more objectively than just getting the top percentage of distributions, but to identify where can we actually find sweeps occurring in this kind of population in these different time periods. So I'm working on that now, and I'm very excited. Hopefully I'll have that to show you in the near future. But in the meantime, what can we say? So what are the consequences of novel pathogen on a host genome? Well, we see a temporary reduction in genetic diversity, and we see almost certainly polygenic selection for resistance and tolerance, although we're still kind of nailing down exactly what that means, how many different loci, what are those different genes involved. And so, kind of, I just wanted to give you a hint at where my research is going forward, in case anyone's interested in collaborating. I mean, I'm just down the coast of California a little bit. So, you know, that was just looking at the House Finch, but one of the things I'm really excited about is to use a system where we can ask this of different species and take advantage of parallel signatures of selection in response to a novel pathogen. And that pathogen is West Nile virus. So in birds, West Nile virus got to the US in 1999, and there are different cohorts of birds that either had a decline and stayed declined, kind of kept declining, had temporary declines, and these are scattered all throughout the avian phylogeny. So it's really a chance to kind of use this power of different related species from kind of throughout the bird tree to understand, you know, are there certain genes or certain regions of the genome that predict which type of response to West Nile these different viruses had. So that's one project I'm excited about. Another project is to work on the many introduced species in Los Angeles, especially this one, pin-tailed whydah and scaly-breasted munia, that provide the opportunities for population genetics and kind of bringing back to the disease perspective, but understand how are these species evolving and when they're in this new place with a whole new community of diseases that they're being exposed to. So I'm excited about that project as well. But anyway, I wanted to just conclude now with my kind of different time scales that I've looked at. What we've shown is that pathogens are one of the most consistent selective pressures across birds, that viruses are shared selective pressures in birds and mammals, that selection in recent time scales and populations likely results in long-term signatures of positive selection, and finally that rapid adaptation likely occurs at many different places in the genome with polygenic selection. So of course had many different people to thank for the many different parts of this talk and funding. Of course museum collections are important for this as well as my color work, especially MVC, yay! And I am happy to take any questions that any of you might have.
So one thing we see in host pathogen coevolution is that both these arms race processes are very important,
Yeah.
but also kind of fluctuating selection and negative frequency dependence is also very important. And it seems to me, so does the DN versus like PN test, is that able to distinguish when you have positive selection for diversity versus not having selection for like directional evolution?
It's not so good at that.
Yeah.
So basically you can do like positive versus negative selection. There are other tests that I'm interested in trying for detecting like frequency dependent or balancing selection, but yeah, that is something that we'll have to do with kind of a different type of test. Yeah, that's a good point, you know, because it really could occur, these long-term signatures could be due to kind of ongoing positive selection or like fluctuating fluctuating frequency-dependent selection. Yeah, good question. Yes?
Yeah, this is just outstanding stuff in my mind, pulling together those different elements of these microbes, you know, interacting with wee gigantic creatures in a whole variety of ways, familiar to us from standard, you know, evolution education of how it's supposed to work and so on, and plugging those in. So I was really pleased to see your progress in that regard.
Thank you.
So let me just nibble on one little thing.
Yeah.
Say, to what capacity, or are you able to say, I know you have some good examples of this, of say, what is the selection rate, and to what extent are you able to detect more subtle selection, or when we say selection and you're looking at the genome, so there's a whole complex, diverse of things, maybe a whole bunch of interesting things are happening, but it's out of sight because it's kind of minor, maybe it adds together to a big effect.
Yeah.
We could fantasize lots of ways things can work, and maybe all those things are working too. It looks like there's enough noise in normal life, you know, through generations to have lots of things happening at once.
Absolutely.
But, so, let's see, I need to make a question out of it. Well, how about the part about what, you know, what extent of selection? It sounded like that Mycoplasma T, you know, did some spectacular things, populations going extinct and so on. You can't get bigger than that, you know. And so, but are you able to detect, say, more subtle things, you think, more subtle levels, or maybe that's, you know, we have to wait a couple generations of our progress to get there. Right now is obviously the time to go for the big names, and my God, you're looking at birds and mammals under one heading. I mean, who could be more satisfied than that? But anyway, I'll just leave it at that as far as the detection of
Like
selection
subtle
levels.
Yeah, absolutely, and I think I have really high hopes for this machine learning method to actually detect these very subtle signatures of selection. It's interesting with the House Finch mycoplasma system, like you think that's a pretty strong selection event, but it still seems it's been really hard to detect what regions of the genome are under selection. So I do think with some of those tools and some of the other tools that are coming out, we'll be able to detect them, but I think also combining these types of scans with more functional types of data, like with more, let's say, I would love to have kind of transcriptomes of experimentally infected house finches, especially from different parts of the range. I think that would go a long way for really nailing down some of these really subtle signatures. Kind of anything you can do to pinpoint some regions of the genome is kind of out of the big, you know, the many millions, and I think I had like 50 million SNPs in this data set, so that of course reduces power a lot. So anything you can do to increase that power will help with those subtle signatures.
And I'll just throw this one other little item in. It seems like right at the beginning you said, oh, the MHC, we all heard of that, let's forget it now and go on to everything else.
Well, I'm not saying we should
so,
forget.
And so if you could add, if you have anything to add about MHC role.
Yeah, MHC is certainly important
in both
too.
birds and mammals, we're talking about a major mechanism, right?
Absolutely. Yeah, so that's hard to get at with these types of selection scans because MHC is so complicated, but it's very complementary to this type of work. And we know in birds and in mammals, MHC is really important in providing protection against certain types of pathogens. So, you know, I definitely think kind of my dream was to do MHC with house finches too, in addition to the genome sequencing through time, and I think that'll be a really kind of valuable contribution to understanding, you know, what are there certain MHC alleles also that might be changing in frequency over time as well, and subject to some of this kind of directional or balancing selection. Yeah. So.
Can you tolerate one more?
Yeah.
Well, what about something just arriving in a population and being able to detect that, and how long it's been there as well is of interest, but in any case, being able to detect it at all, and where we anticipate, just like I think you had some examples near the end of things you might go after in that regard. Something new, West Nile virus, I think it was.
Yeah.
And, how is this going to be introduced, and how will we detect this? And so I think that's quite intriguing, and maybe even thinking about human populations as well.
Absolutely.
Down the road, maybe not that far down the road.
Yeah, there's a lot of kind of, could be good tie-ins to modeling, once we're understanding more of these evolutionary dynamics, like building those into models to estimate how species might respond to new different types of diseases will be really valuable contributions as well.
And we have this whole, irritation of the medical system, which means people don't die, they can even reproduce and so on. They get sick, and, that would ordinarily in the old days, they would not be there anymore, but now we don't have that. And so that's kind of a complication in some ways, but it also is something that is happening in real life, to be sure, and humans get away with a lot of malarkey. Probably most of us in our own lives, maybe we shouldn't be here, because of that. And so it's kind of another layer to keep in mind. We may not be looking at strong selection effects, but there's actions that could have been selection in the old days easily. I mean, invasives of one sort or another. And just one last.
I think we have to move on to a little bit last question, and
then
then
the very last.
then we.
I love that part about viruses coming out on top. I think I always felt they were the big guns that larger creatures had to deal with. They're so damn fast. I mean, how could we ignore that? Thank you very much.
Last question, Libby.
I was just going to ask you think that your machine learning method, where you're using all the different kinds of approaches, are going to pull out non-coding regions?
Highly possible. Yeah, so that's, another caveat is I'm almost exclusively focused on coding regions here, but I think that there's a lot to be said about non-coding elements as well. So
yeah, that could be really exciting to tie with the transcriptomic.
Absolutely. I mean, we know there are differences in gene expression, so yeah, I'm excited to dig into the non-coding stuff for sure.
All right, let's thank Allison for a great talk.